Good morning friends. Today I'll be discussing part 2 of cancer. In this section I'll be dealing with oncogenes and tumor suppressor genes. Now what are proto-oncogenes? Proto-oncogenes are regulatory genes. The products of many of these proto-oncogenes are polypeptide growth factors. For example, the cis gene produces PDGF or platelet-derived growth factor, which is required for normal wound healing. The products may also act as receptors for growth factors. For example, the ERB gene produces receptors for the epidermal growth factor. Now, proto-oncogenes are also under the control of regulatory genes and expressed only when they are required. When a virus enters, an extra oncogene is inserted. So, there is a continuous expression of gene leading to the uncontrolled cellular activity and malignant transformation. So, proto-oncogenes are normal cellular genes that affect growth and differentiation. Since they are cellular, it is represented as C capital onc, that is cellular proto-oncogene. One of them is, so to express the SRC gene, which is cellular or a proto-oncogene, you express it as CSRC. Now what are oncogenes? Oncogenes are genes whose products are involved with neoplastic transformation. So they can be brought about by viruses, and then therefore it is represented as VONC. VONC, that is the viral gene. And the viral gene or the oncogene of SRC is known as the VSRC or the sarcoma producing gene. Now the oncogenic products, what are they? One example is the protein tyrosine kinase. Greater than 50% of oncogenes produce this enzyme, and this causes abnormal phosphorylation. Secondly, you have a receptor-associated tyrosine kinase. The receptors for certain growth factors, such as the epidermal growth factor, the myeloid growth factors, are transmembrane proteins with tyrosine kinase activity. Third type is the GTP binding proteins. This is seen in the case of the RAS oncogene, which produces GTP binding proteins seen in human cancers. Nucleoproteins, the products of the MYC gene, the FOS gene, and the MYB gene, oncogenes are nucleoproteins which modify cell growth. So the main type of oncogenic products which are produced by these oncogenes could be the protein tyrosine kinase, or it could be receptors of certain growth factors which have tyrosine kinase activity. Third, they could be GTP binding proteins, as in the case of the RAS oncogene, or it could be nucleoproteins like the products of MYC, FOS, MYB oncogenes are all nucleoproteins which modify cell growth. Now if you take a virus like the Rao sarcoma virus, the genome of this retrovirus contains 4 genes. So this virus contains actually 4 genes. It's a retrovirus. Okay. First gene is the GAG gene, which codes for group specific antigens of the virus. Second is the POL gene, which codes for reverse transcriptase. Third is the ENV gene, which codes for, or the envelope gene, which codes for glycoproteins of the viral envelope. And lastly, we have the SRC gene, or the sarcoma causing gene. Now the product of SRC is a protein, that is the enzyme tyrosine kinase, which is found to be responsible for the transformation, okay, the cell transformation. So this Rao sarcoma virus, it contains 4 different genes, but the sarcoma causing gene is the SRC gene. And this product of this SRC gene is an enzyme tyrosine kinase, and it is responsible for transformation, malignant transformation. Now, growth factors. The growth factors are usually polypeptides. They can initiate cell migration, differentiation, remodel tissues. It is involved in various stages of wound healing. It affects different types of cells, like it can affect the epithelial cell, the nervous cell, or the mesenchymal cell. It can regulate stem cells, that is, it can make them to differentiate and form different types of mature cells. The mode of action of these growth factors are endocrine, like hormones, and paracrine, that is, it affects the neighboring cells, autocrine, that is, affects cells which secrete them. So its mode of action could be endocrine, paracrine, or autocrine, and they have high affinity receptors on the plasma membrane of target cells. Now, this table gives us an idea of the examples of oncogenes or proto-oncogenes, what are its oncogenic products, and what is the associated cancer with this gene. So very often you are asked a question on give examples of oncogenes or proto-oncogenes and examples of the products and the type of cancer that it causes. So when you look at the cis oncogene, the oncogenic product is PDGF or platelet derived growth factor. The type of cancer it causes is osteosarcoma. The HST1 gene, the oncogenic product, is the epidermal growth factor, and it can cause cancer of the stomach, the breast, and the gallbladder. Now we look at the next type, that is where the oncogenic product is not the growth factor but the receptors for the growth factors. Under this, we have different types of oncogenes. For example, ERB-A and ERB-B1 is responsible for producing the receptor, that is for transforming growth factor receptor. ERB-A is responsible for causing colon cancer, and ERB-B1 is responsible for causing lung cancer. Now, ERB-B2 produces the epidermal growth factor receptor, the second type, EGFR, and ERB-B3 also causes an epidermal growth factor receptor. ERB-B2 is responsible for stomach cancer, and ERB-B3 is responsible for breast cancer. Now, these proto-oncogenes also produce signal transducing proteins like GTP binding proteins or the tyrosine kinase enzyme. So, the RAS gene is responsible for the oncogenic product as a GTP binding protein or a G protein. The type of cancer it causes are the leukemias, lung cancer, pancreatic carcinomas, and colon cancers. The ABL gene releases the oncogenic product tyrosine kinase, and this is responsible for causing leukemias. So this table gives you in a nutshell what are the oncogenes or proto-oncogenes, what are the main oncogenic products, which I have grouped as those genes which produce growth factors as its oncogenic products, those genes which produce growth factor receptors, and those genes which produce signal transducing proteins like the GTP protein or the G protein or the enzyme tyrosine kinase. Also, I have given the cancer which it causes. For example, this is gene causing osteosarcoma. Now, proto-oncogenes have to be converted to oncogenes for malignant transformation to occur. So, there are 5 mechanisms by which proto-oncogenes are converted to oncogenes. These 5 mechanisms I have listed here. The first is promoter insertion, second is enhancer insertion, third is chromosomal translocation, fourth gene amplification, and fifth point mutation. Let's look at the first of these 5 different mechanisms, that is promoter insertion, whereby proto-oncogenes are converted to oncogenes to produce malignant transformation. Now, certain retroviruses like the avian leukemia virus lacks a oncogene, and they may be only able to cause cancer over a long period of time as compared to retroviruses which contains the viral oncogene. Now, when such viruses like the avian leukemia virus infect cells, a copy DNA is synthesized and it is integrated into the host genome, and this is called the provirus. This provirus is flanked by long terminal repeat sequences. These act as promoters as it is inserted upstream, and transcription and translation of the micRNA occurs. Now, on the left side, you see the MYC gene. This is an inactive MYC gene of normal chicken B lymphocytes. It is in an inactive state. Okay. Now, what happens? It is infected with the avian leukemia virus. When it is infected with the avian leukemia virus, it produces a copy DNA, and this copy DNA is known as the provirus. This gets integrated into the host DNA, and it is flanked by, as you can see, long terminal repeats, both on the left side and the right side. So the copy DNA, which is flanked by long terminal repeats, acts as the promoters, resulting in the transcription and translation of the micRNA. Okay, and this can result in malignancy. Okay, so the first type is promoter insertion. The promoter in this case is the provirus, which has got inserted into the host genome, and it is flanked by the long terminal repeats, which then acts as promoters, resulting in the activity of the micRNA. That is, it then goes on to for the process of transcription and translation. So it enhances the activity of the micRNA. In the second case, we see enhancer insertion. So in some cases, the provirus is inserted downstream. It is not inserted upstream, but it is inserted downstream. So in the in the figure, you can see on the left side, you have your inactive MYC gene. It is not undergoing any transcription or translation. It is not giving rise to any type of malignancy. Okay, it is in an inactive state. Now what happens? It is infected with the avian leukemia virus, and the avian leukemia virus produces a copy DNA. This copy DNA, which is also gets inserted not upstream, but it is inserted downstream from the MYC genome. Okay, so it cannot act as a promoter. However, certain sequences, again it is flanked by long terminal repeats, and certain sequences of the provirus now acts as an enhancer element and activates the upstream MYC gene, leading to its transcription and translation. Okay, so promoter insertion is done. Where is the promoter insertion done? It is done upstream to that of the MYC gene. So it can act as a promoter. The provirus acts as a promoter and it activates the MYC gene. But in the second case, the provirus is inserted downstream from the MYC gene. So it cannot act as a promoter, but certain sequences acts as an enhancer element and activates the upstream MYC gene, which is leading to its transcription and translation. The third mechanism is known as chromosomal translocation. Now, most tumor cells shows chromosomal abnormality. For example, Burkitt's lymphoma, which is a fast-growing cancer of the B lymphocytes. In this case, a piece of one chromosome is split off and joined to another chromosome, and this is known as direct translocation. So you find that one chromosome from chromosome 8 is broken and it is joined to chromosome 14. Okay, and this process is known as direct translocation. So what is happening here, that is a piece of a chromosome from the Q arm of chromosome 8 is broken off and moves to chromosome 14. A small segment of Q arm of chromosome 14 breaks off and moves to chromosome 8. So there is an interchange of a small segment of the chromosome. One segment of the chromosome from the Q arm of chromosome 8 breaks and moves to chromosome 14, and a small segment from the Q arm of chromosome 14 breaks and moves to chromosome 8. So in this particular case, you can see that the MYC gene from chromosome 8, which is in an inactive state, is translocated to chromosome 14. Next to the gene, that is the H-chain gene, which is transcribing for immunoglobulins. When this is translocated adjacent to the H gene, which is actively transcribing for immunoglobulins, the MYC gene becomes activated. It then goes through the process of transcription and translation, and this process is known as reciprocal translation. So this figure very elegantly shows the type of reciprocal translocation. We find that the MYC gene, which is residing in the chromosome 8, that the Q segment of chromosome 8 is cut or it's split and it gets shifted to chromosome 14. Simultaneously, a short segment of chromosome 14 is split off and it joins the chromosome 8. So in the second half, you find that chromosome 8 now contains the blue portion is the small segment of chromosome 14, and chromosome 14 has the small segment of MYC C, okay, lying adjacent to the gene, H gene, which is actively transcribing for immunoglobulins. Now, since it is placed adjacent to a gene which is actively involved in the formation of immunoglobulins, that is, it's actively transcribing and forming the proteins, in this particular case immunoglobulins, the MYC gene, which was inactive on chromosome 8, has now got activated and goes through the process of transcription and translation. So this process is known as reciprocal translocation, because there is an interchange, reciprocal interchange. A segment from 8 goes to 14 and a segment of 14 goes to 8. And in this process, the MYC gene, which has been translocated in this way, becomes activated. The fourth mechanism is gene amplification, which is seen in certain tumors. So this can be detected as homogeneously stained regions on a specific chromosome. Now we find that the amplification of the N-MYC from 3 copies to around 300 copies is seen in neuroblastomas. And when such amplification is detected, then we know that the prognosis is very poor. Gene amplification is another process of malignant transformation, that is the conversion of the inactive N-MYC, which is responsible for neuroblastomas once it is activated, and many copies of it is formed by gene amplification. And such amplification, when detected, will indicate a very poor prognosis for the patient. And lastly, we have single point mutation. Now, in the case of the murine retrovirus oncogene, the viral RAS produces a protein known as P21. This modulates the activity of adenyl cyclase and cyclic AMP. Now, the proto-oncogene of CRAS, that is the cellular RAS from normal human cells, and the VRAS of oncogenes from cancer, they find it's different only in one base. So, a point mutation has occurred to convert it to the viral oncogene. Okay, from the cellular oncogene, which is inactive, that is the proto-oncogene from the cells, that is the CRAS, to convert it to the VRAS, which is the active oncogene, you find that it has undergone a point mutation. So, single point mutation is another mechanism for converting a proto-oncogene to an oncogene. Now, this sort of a mutation results in diminished activity of the enzyme GTPase, increased activity of enzymes like adenylate cyclase and cyclic AMP. This causes altered cellular metabolism and causes malignant transformation. What are the biochemical changes we commonly see in cancer? There is increased replication and transcription, increased synthesis of DNA and RNA. That means increased synthesis of DNA and RNA. Secondly, there is increased glycolysis. Why is there an increased glycolysis? Because there is increased energy demand. Both aerobic and anaerobic glycolysis is noted. There is synthesis of fetal proteins like carcinoembryonic antigen and AFP, and they form, as you know, very important tumor markers. The cell surface glycoproteins and glycolipids are altered. Then we can see secretion of certain proteases, protein kinases. Protein kinase is an important oncogenic product which can be detected like tyrosine kinase. There is appearance of new antigens. Like for example, carcinoembryonic antigen is a very unique antigen which is detected in certain types of tumors. And there is inappropriate synthesis of certain hormones. Certain hormones become extensively synthesized, and so also certain growth factors. Next we come to tumor suppressor genes. Tumor suppressor genes produces a protein product that normally suppresses cell growth or cell division. So, as you can see in the diagram on the left side, what is the action of the tumor suppressor gene? It is producing a normal growth inhibiting protein which is controlling cell division. So, it regulates cell cycle and apoptosis. So, it is also called as anti-oncogenes. Now, what happens if a tumor suppressor gene is altered by mutation? This results in the fact that it is not being able to produce that growth inhibiting protein. So, the loss of inhibitory effect of product. What is the product? The growth inhibiting protein is not formed. And this results in inactivation of growth control mechanisms. And so, it causes some type of tumors. Now, two very important tumor suppressor genes are the retinoblastoma gene or the RB gene and the P53 gene. So, tumor suppressor genes, they produce a protein product, as you can see on the left hand side of the figure, which normally controls cell growth or cell division. So it is regulating cell cycle and apoptosis. They are also known as anti-oncogenes. Now what happens if this becomes mutated? The tumor suppressor gene becomes mutated. So the normal product, which is a growth inhibiting protein, becomes defective. And what happens? You'll have uncontrolled growth. Cell division is not under control. And this results in certain types of tumors. So the RB gene or the retinoblastoma gene and the p53 gene are two examples of the tumor suppressor gene. What is the retinoblastoma gene? This was the first tumor suppressor gene discovered. Now the mutation can lead to several human tumors, and most commonly it is the retinoblastomas. This tumor suppressor gene is located on chromosome 13q14, and this is mutated in many forms of cancer. The growth inhibitory phosphoprotein it produces, the product of the retinoblastoma gene, which is a growth inhibitory phosphoprotein, is known as the PRB protein. Now, the other very important tumor suppressor gene is the p53 tumor suppressor gene. This gene is located on chromosome 17 and prevents multiplication of genetically damaged cells. When the DNA of the normal cell is damaged by carcinogenic agents, it causes activation of the p53 protein. Now, the product of p53 protein is a nuclear phosphoprotein of 53 kilodaltons. The p53 will bind to specific sequences in the double-stranded DNA and act as a transcriptional regulator. It can cause inhibition of mitotic activity by binding various viral proteins and inhibiting cell division. It prevents the cell to enter the G1 phase transiently, and it allows repair mechanisms to operate. It also causes the promotion of apoptosis, that is, it identifies the genes with damaged DNA. If it cannot be repaired by inbuilt systems, the p53 directs such cells to apoptosis. If p53 is inactivated, large amounts of tumors can occur. So p53 is known as the guardian of the genome, or it is known as the molecular policeman, or it is known as the gatekeeper gene. So there is a functional balance in the normal cell between the oncogene and the tumor suppressor gene. As you can see in the first figure, there is a balance between the oncogene and the tumor suppressor gene. When there is a functional imbalance and you have more of oncogenes and less of tumor suppressor gene, then it can cause cancer. Okay, this leads to cancer. So the imbalance of oncogene and tumor suppressor gene can lead to the formation of cancer cells. That is why p53 is known as the guardian of the genome, it is known as the molecular policeman, or it is known as the gatekeeper gene. So let's look at the function of the p53. Due to various environmental factors, cells can get damaged. There are two options. The damage to the DNA may be minor and can be repaired. For this, the cell activity is halted transiently, the gene function is suspended, the gene is repaired, and once the cells are repaired, it goes back into action. The other is there is severe damage to the DNA. So it cannot be repaired, and such cells are then directed for apoptosis. So this figure very elegantly shows what is the role of the tumor suppressor gene. As I told you, in the normal gene, the tumor suppressor gene is regulating cell growth. Okay, so it is like, a car moving with, with, with all its brakes and accelerator all under control. Okay, now what happens? You have the first mutation. All right, so this becomes the gene becomes a susceptible carrier. All right, so now what happens? With the first mutation, an active oncogene has got incorporated. All right, with the second mutation, what happens? There is both the active oncogene as well as which is proliferating, and there is also a mutation of the tumor suppressor gene. Okay, so what happens? What is the effect of the oncogenes? The effect of the oncogene is like putting your foot on the accelerator. So more it proliferates, the more speed it will gather. As long as the tumor suppressor is there, it will act as a brake. Okay, but if the tumor suppressor gene is mutated, then there is no brake. So what happens? It leads to the crashing of the car, or it can lead to what can be described as the cause of causing cancer. So the active oncogene is allowed to proliferate without any brake because the tumor suppressor gene has got mutated, no brake. So this will be totally, your foot is on the accelerator, leading to active proliferation of the tumor cells. Thank you.